between the Italian and the French Society of Parasitology. Last year, we organized the first meeting and we presented the two societies and also the different topics and the different events by the two societies. And I think that since then we are even strengthening our collaboration in different projects and research together. And with the Gregory, with Maria Paola, and with Adriano Casulli, we set up this second meeting, and we chose two different topics that could be of interest, so artificial intelligence and eDNA. And I give to you the word, Gregory, and then we can start, I think.
Yes, thank you, Laura. It will be very quick. So I thank you, Laura, Maria Paola, and Adriano, and of course all participants for attending this webinar, the joint webinar. And I think, Laura, you said everything. We have strong collaborations. So no, I think with your society, and of course we will continue on both scientific projects, but also on more administrative stuff. For example, for these journals we spoke about. And today we decided to have a more maybe technologic approach. So these two sessions will be very interesting for both of you, SFP and Soipa, I think. And I hope you will all enjoy this meeting. And okay, I will try to find.
Yes, I've heard the program, so maybe I can introduce the first
Yeah,
speaker, who is from the French Society of Parasitology, Cécile Nabet, who is going to talk about AI and
Hello.
diagnosis in medical entomology, focus on MALDI-TOF mass spectrometry. And Gerard, if you agree, I would put the discussion at the end of the three presentation of the first topic on artificial intelligence. Okay?
yeah, sure.
So please, Cecile, you can share your screen.
Thank you. Hello, everyone. I'm very happy to be here at this second joint webinar between French and Italian societies of parasitology. I'm an associate professor in parasitology and mycology at Pitié-Salpêtrière Hospital and Sorbonne University. And we are all aware that the global changes are reshaping the way vector-borne diseases spread all around the world, and these changes can favor the introduction of invasive species, the expansion of local ones, and even modify their competence as vectors. So for effective prevention and epidemic preparedness, we need efficient real-time surveillance tools to monitor blood-feeding arthropods. So what are the key parameters that we can monitor? Species identification is essential, but it is not enough. So to better understand and control vector-borne diseases, we must also monitor biological threats directly linked to transmission dynamics, including survival, vector competence, pathogen infection status, blood meal history and preference of host, and also, for example, the susceptibility to insecticides. So by combining species identification with these biological traits, we can move toward better informed control strategies. And that brings us to the toolbox for the characterization of the vectors. Traditional entomological methods like morphological identification are reliable, but they are slow and they require taxonomic expertise. Molecular methods are more accurate, but remain expensive and time-consuming for extensive screening. So that's why numerical methods that are alternatives are emerging, like computer vision, sensors to detect wing beat frequency, infrared spectroscopy, and also mass spectrometry are being developed. They are aimed at faster, automated, large-scale surveillance of arthropods. So how can the MALDI-TOF tool revolutionize vector surveillance? So this method is based on protein profiling. That means separating proteins on their mass to charge ratio. This generates unique spectral fingerprints, characteristic of a species, strain, or physiological state. And this is already a revolution in clinical microbiology because this approach is becoming now a gold standard for fast and robust identification in laboratories for the identification of bacteria, fungi, and parasites. And it is now showing great promise also in entomology for the characterization of arthropods. So how does Malditov mass spectrometry works? In practice, arthropods are dissected to select the anatomical part of interest, and usually it is the legs, the thorax, or head. Then proteins are extracted and deposited on a metal plate and covered with the matrix. When the laser hits, proteins are released, accelerated through a flight tube, and detected according to their mass. The produced mass spectrum is compared to a reference spectra database. Identification is achieved through pattern matching algorithms that compare peak position, ranking the closest match. So the sample can only be identified, of course, if there is a matching reference in the database, which brings us to one of the biggest challenges of the use of Malditov. And to address this gap, we have begun integrating arthropod databases into the MSI online platform developed at Sorbonne University and Hospital of Paris. Initially created for microbial identification, this platform is an open access platform where researchers can share mass spectral databases and now also arthropod spectra. So the goal is to create the same openness we have with molecular sequence databases, but for Malditov. And now the question is how we started with artificial intelligence. While we were implementing arthropod databases into MSI platform, we have noticed something unexpected in Anopheles mosquito spectra from Mali. The spectra were not randomly distributed, they were grouped according to physiological state. And as you can see in this dendrogram, which groups the spectra depending on their protein profile relatedness, we have all the visually blood-fed specimens that belong to the same cluster. So this observation triggered an important question. Can Malditov be used not only to identify species, but also to assess mosquito biology and key determinants of pathogen transmission? More specifically, can we identify, for example, the age? Can we detect the blood meal history or the Plasmodium infection? At that time, machine learning, particularly supervised learning approaches, had already shown their power in improving the discrimination between closely related species and also to distinguish microbial strains according to their antimicrobial resistance profiles. In supervised learning, we start with labeled data, meaning we know the biological status of each specimen, and the algorithm then builds its own parameter of classification by adjusting mathematical parameters based on the training data. To ensure robustness, we need a validation dataset to prevent overfitting, in other words, to avoid simple memorization. A test dataset is then used to evaluate the performance and assess how well the model generalizes to new unseen data. So at that stage, importantly, while machine learning had been used to improve species discrimination in mosquitoes, it had never been applied to study their biological traits. So we started working closely with computer engineers. We explored whether neural networks could detect subtle spectral patterns linked to the biology of Anopheles mosquitoes. And our goal was to predict key biological parameters to vector control the age, the history of blood meal, and the Plasmodium infection. To test this, we used a controlled experimental model, a laboratory colony of Anopheles stephensii infected by Plasmodium berghei strain. Mosquitoes were collected and dissected at different time points after blood feeding, allowing us to generate well-labeled data sets. And we evaluated convolutional neural networks, CNN, a type of artificial neural networks that is particularly effective at detecting complex patterns. So what did we observe? When we examined the spectra visually and by protein profiling, there were no obvious specific peaks distinguishing one biological category from another, but instead what we observed were variations in peak intensity, reflecting subtle proteome changes. And this is precisely where our artificial neural networks showed their strengths in this study. The models were sensitive to these proteomic variations and successfully recognized spectral patterns associated with the mosquito age, 73% of mean accuracy, blood feeding history, 89% of accuracy, and Plasmodium berghei infection, 78% accuracy. So why this matters? This proof of concept opened a new field of research, a new avenue. For the first time, we demonstrated that Malditov coupled with AI can do more than identifying Anopheles species. It can also characterize biological traits directly linked to malaria transmission dynamics. But of course, an important question remained. Does this still work under real-world conditions? For example, what happens when we analyze mosquitoes collected in the field with genetic variations, ecological variability, and transport constraints? And to begin answering this question, we focused on one key parameter, which is mosquito age. And we collaborated with the University of Dakar in Senegal. We focused on the species Anopheles arabiensis. The specimens were collected at the larvae stage from two ecological sites in Senegal, located 205 kilometers apart, in order to introduce genetic diversity and environmental diversity as well. The mosquitoes were reared in an insectarium and sacrificed at different ages to build a labeled dataset. And in this study, we tested new deep learning approaches and models, including temporal convolutional networks, which is a model that is particularly effective for the temporal dependencies analysis. So instead of classifying the mosquitoes into age groups, we used a regression approach, allowing us to predict age as continuous values in days. These figures that you can see for the two sites highlight the high level of correlation between the actual and the predicted age. And despite a relatively small data sets, 183 specimens from one site and 68 for the other, we achieved an age estimation accuracy of 1.7 to 2.6 days. And even more importantly, the models were generalizable. That means that a model trained on one site performed well on the other without specific training, with an error of 3 to 4 days. So why is this important? The precision we achieved is highly relevant because from an epidemiological perspective, because an Anopheles mosquito typically become capable of transmitting malaria from 11 to 16 days after infection. It means that it includes the time for the mosquito to get an infected blood meal and also the time of incubation of the parasite. So an estimation error of about 2 days is precise within this transmission window. And notably, the mosquitoes were originated from the field with limited sampling effort. So why does this matter for malaria control? Because current age grading methods are approximate and time consuming. For example, the ovary dissection to estimate the parity, which is actually the reference in the field. Mosquito survival is a key determinant of malaria transmission intensity. And improving the age estimation can enhance epidemiological transmission models and also the evaluation of vector control strategies aimed at decreasing the survival of the mosquitoes, especially the insecticides. So some questions are remaining. What are the performance on mosquitoes collected at the adult stage and the reproducibility on other mosquito populations and species? We are now pushing the boundaries with new machine learning models. For several years, we have been collaborating closely with computer science experts, exploring different model architectures. And we have improved the accuracy for the species of the Anopheles gambier complex. We have reached 85% accuracy for the discrimination, and we have also confirmed the robustness of age estimation. And now we are working at integrating these models into the MSI platform. The initiative is growing. We are extending the approach to new arthropods, for example, the ticks, the Hypobosidae, and the Dermanicidae. So now we are moving from a proof of concept in mosquitoes toward a broader integrated tool for vector ecology. So in conclusion, Malditov coupled with artificial intelligence opens the door to entirely new applications in entomology. Its potentials goes far beyond simple species identification. It allows us to explore biological traits directly linked to vector competence and pathogen transmission. Fast, robust, and adaptable, this approach is particularly well suited for automated large-scale monitoring of arthropods. And with the development of the MSI platform, we now have the opportunity to transform this innovation into a shared, collaborative surveillance tool for the scientific community. Thank you for your attention, and I would like to especially thank Professor Pierre Roux and Anne-Cécile Normand, who were at the initiative of the MSI platform. Thank you.
Thank you, Cécile. Thank you very much also for being in time. And I'm sure there will be a lot of questions and curiosity around this nice job. Thank you very much. So maybe we continue. Where is Greg with the next speaker?
Yeah.
Maybe, yeah, Greg, you want to, yeah.
So yes, Cécile, thank you very much. It was really interesting. Each time I hear you, it's so interesting. And now I will give the floor to Noemi Sarletti, who will speak about artificial intelligence for entomological research and surveillance on mosquitoes, including vectors on the transmission of borne pathogens. And it was a Mosaico project.
Hey, good morning. Good evening, everyone.
Thank you.
I share the presentation. Okay. You can see the slide?
Yes.
Okay. Okay. So, good evening, everyone. Let me introduce myself. My name is Noemi Serletti, and I am a PhD student. I work at Istituto Superiore di Sanità in the working group of malaria in the Department of Infectious Disease. Today, I'm going to talk about Mosaico project. Mosaico is an acronym that means Mosquito Artificial Intelligence Control. In the following slide, I will show you the results that we have obtained during these 3 years. But first, let me start by giving you some background information about this project. According to several studies, the traditional method of mosquito surveillance is a labor-intensive and time-consuming process because to complex operations, in particular rapid identification of mosquito species after field catches that generates non-homogeneous data across national territory. Today, artificial intelligence and computer vision technologies are promising tools to address problems in object detection and object classification in general. And in fact, the Mosaico project, in collaboration with several partners, uses these new techniques for adult mosquito identification, in particular for autochthonous species, which play a relevant role in public health, but also for possible invasive species such as Aedes aegypti, allowing an automation, standardization, and scaling up the actual surveillance activities. Our ultimate goal is to develop an automatic mosquito detection device that is able to take high-resolution multi-object images and to perform in real time an object detection thanks to a pre-trained deep learning algorithm classifying at genus and species level each individual mosquitoes in these multi-object images. We have implemented this idea by creating a prototype that can do all of this. In detail, the present Mosaico prototype is composed by two distinct components: a local part or imaging system to obtain the high-resolution multi-object images and includes the Mosaico device, so the hardware of the system, and the associated app to remotely control it. The capture images are transmitted to a dedicated server that is the remote part recognition system. The server is located at Istituto Superiore di Sanità. It contains the deep learning models that process the image in a few seconds, generates an output. These model responses are transmitted to the hardware, to the Mosaico device, and can be viewed on the app in real time. So, in other words, our system takes a picture of several mosquito, actually 82 mosquito specimens from a single image, and predicts the species to which they belongs. This prediction is made using a set of species on which the deep learning model has been trained. In our case, 7 species, and in the following slide, I will show you the reasoning why actually we have only 7 species. The next video shows the entire operational workflow from the loading of samples to models output. This is an overview of a Mosaico device, and the step to use it is very simple. The first step is to put inside the device this special grid with mosquito samples from field catches, and at the same time turn on the camera and open the app on personal PC. Log in the app with a specific username and password, and switch on to the remote camera. Before, check that the settings are corrected and the image is in focus, and take the picture. Before to send this picture to models, to deep learning model, the operators fill the window with information about the field catches. For example, the name of trap, the geographical coordinates. Also, you can see these geographical coordinates on Google Maps directly by this window. And other information, for example, sample type and sampling type. Submit the image in a few seconds, you have this type of output. So, the model responses, this model output, are associated with an estimate of classification confidence, which is itself a function of the accuracy of classification process. This confidence estimate is visually represented to the user with a system of colors. A green box with the name of the best matching species inside it appears when the system is highly confident that in its identification, while a yellow box appears when the model is unsure about its identification and in fact suggests two possible species among the 7 species used in the training. And finally, a red box appear when the model is not confident that the mosquito inside this box belongs to any of the 7 species. And a red box is caused by autochthonous species, but the specimens in particular is highly damaged or the species is not included in the training set, but also by a new species, so an invasive species. This red box is a visual warning to operators, prompting them to undertake more analysis on these specimens. Now let me go through the steps of prototype development, and the first step is the collection of specimens. In fact, the acquisition of training data is of critical importance in order to obtain a deep learning model like this. And then, thanks to a national network of insectary fertility that was created in the frame of PNRR project, the Mosaico National Network was created with 18 partners from 16 regions. The partners were involved in the collection of mosquito samples from their colonies in the year 1 of the project and from field catches during the surveillance activities in the year 2, with the aim to train, validate, and test the deep learning model. The species to be collected have been selected for being medically relevant, so primary or secondary vector of transmission bird pathogens, or for being geographically abundant, so likely often present in mosquito surveillance activities, so in the drops. At the end, only 7 species have been used so far for algorithm training, and 2 parameters determined this choice. The first parameters, the most important parameters for a deep learning model, is the number of specimens available for each species, and second, their origins. At the least 3 independent geographical sites for each species. The results that we have obtained using the mosquito specimens from Mosaico network in these 2 years show that the algorithm identify with high confidence level the 7 species analyzed. In fact, we can see along the diagonal of the final confusion matrix the portion of correctly classified values, so the accuracy score in mosquito classification. The accuracy range is between a minimum of 80% to a maximum of 97%. In our training set, there are 2 similar species, Aedes albopictus and Aedes aegypti. And in fact, the most important difference between these species is located on the thorax. Aedes albopictus has only 1 white line, and Aedes aegypti has many lines in the shape of lyra. Despite the presence of these 2 similar species, our deep learning model is not subject to the occurrence of false positive, and this is most important in order to avoid false alerts on Aedes aegypti in our country. The way of deep learning model works is obviously being improved with a particular attention to the differentiation between these 2 species, but we have also started an investigation about the reasoning behind the few incorrect predictions made by the algorithm using a particular approach, the explainable AI methods, with a technique called GradCam that produce a heat map like this, showing which parts of an image are most important for the final model prediction. Before to use the mosaico prototype in the field, a workshop was held at Istituto Superiore di Sanità with the entire mosaico network in the summer 2025 before the surveillance activity. So on May 8 and 9 May 2025 to train on the use of mosaico device prototype, but also to distribute 10 copies of this prototype. The copies were used in field tests in more 50 sites nationwide in the same summer. And to date, a total of 24 multi-object images, probably actually the images are more in the dataset, but at the end of the summer, 24 multi-object images were uploaded by operators and processed by deep learning model, and the model correctly classified more than 82 mosquitoes and making only the 30% of errors. So this is a very promising result for the future of the project. Meanwhile, the Mosaico team has developed also a Mosaico website where the operators can be viewed the Mosaico, the original image from app and also the model's output, and it also can be downloaded this information, but at the same time the operator can be provided feedback correcting any classification errors made by the algorithm during this validation phase. This was most important for the future, so for algorithm improvement in the future, helping to the Mosaico project goal to improve the national capacity in modern entomological surveillance. This last video show the Mosaico website and what an operator can do in this Mosaico website. To use Mosaico website, it's necessary a login with the same username and password used for the Mosaico app. Inside the Mosaico website, there are a history about the photo that has been taken in with the app. Chose one of this photo, and you can download the original image and the model's output. Moreover, you can leave a feedback in a simple way with a double click on the mosquito of interest, like this. And finally, also you can download a report, also a summary of results about this image as an Excel file. And in this Excel file, there is the operator's feedback. So not the errors, but the operator's feedback. So, in conclusion, Mosaico project tried to introduce innovation in the field of entomological surveillance through the following means. High resolution multi-object images. So actually from a single image, you can identify in a few seconds 18 mosquito specimens with thanks to a doc deep learning model. This model has been developed for Italian mosquito species, but the hope that is this deep learning model can be used also for European mosquito species, and thanks an interactive website, but also a simple app. This prototype can be used by also non-expert personnel. There are so many future steps, I know, but in general, we want to improve the model performance with new species and new specimens, and we want to use the prototype in national surveillance and routine national surveillance, but also in a particular type of surveillance, such as at points of entry for new species, for invasive species, and in area where no data is yet available, such as the south of Italy. And thank you for the attention.
Many thanks, Noemi, for this very, very interesting talk. I think looking at the two first talks, we could imagine maybe an integrative approach between morphology, MALDI, maybe other, but yeah, I think integration could be very, very interesting. Laura, do you want to introduce the last speaker of this session?
Yes, also because I know her for many years, so it's my pleasure to introduce Professor Stefania Zanetti from the University of Turin. And we move on another topic. So, AI-based approach to wildlife pathogen surveillance in integrated health monitoring frameworks. Vai, Stefania.
Thank you very much. Thank you, everyone. Let me share my presentation that should be coming up. Can you see it?
Yes.
Fantastic. So, well, once again, thank you very much for the opportunity to present our experience in applying artificial intelligence-based approaches to improve surveillance of infectious pathogens, with a particular focus for today on parasitic infections in wildlife. The work that I will be presenting has been developed within the research group led by Ezio Ferroglio at the University of Torino and within a broader collaborative network, including INETWILD Consortium and the European Observatory for Wildlife. Before addressing the main methodological aspects, I would like to briefly introduce the conceptual framework that underlines our work, that is integrated wildlife monitoring. For integrated wildlife monitoring, we define the monitoring system that combines active and passive disease surveillance with structured wildlife population monitoring. Its objective is to make wildlife surveillance truly informative for risk assessment and for proactive disease management. This integration is particularly necessary when we deal with wildlife epidemiology because while epidemiological parameters, such as the prevalence, often rely on very precise data. For example, for the numerator, here you see the fraction of the prevalence. We have very precise data in terms of number of animals that are infected with a given pathogens, but almost always, unfortunately, we have very poor data for the denominator. When we want to know, for example, the total population at risk for a certain disease. Without robust estimates of wildlife abundance or wildlife density, even a simple epidemiological parameter like the prevalence is very difficult to interpret and to compare across space, so between different study areas, for example, and over time. Within the integrated wildlife monitoring framework, we developed some standardized methods to generate reliable population metrics, including density and abundance and the community composition of wild species that are relevant for epidemiological purposes and for health risk management. The tool developed within the INETWILD framework to obtain harmonized density estimates is the random encounter model. Random encounter model is a well-established method to calculate, to estimate density of unmarked individuals. It uses camera traps, of course, and it produces reliable and comparable outputs across different study areas and over time. Since it is based on camera traps, it can be applied simultaneously to estimate the density of many different species. Everything that passes in front of the camera trap can be estimated. Of course, it is a technical limiting factor the body size of the animal. For very small species like small mammals or rodents, this approach is not reliable, but for everything from mesocarnivores to large ungulates, the random encounter model is a very promising tool. The methodological details of random encounter model are extensively described in the literature, so I will not go into technical depth, but very briefly, when an animal enters the field of view, the blue area in front of the camera traps, the camera is triggered to burst a high number of images. These images are used to calculate a number of parameters that are necessary for the random encounter model to produce a density estimate. Traditionally, we used to do this process by hand on millimetric papers to estimate the travel distance that you see here in the image of the animal, putting together the information from each individual image. As you can imagine, it was a very time-consuming process, and it was really the bottleneck for large-scale implementation of integrated wildlife health monitoring. This is where artificial intelligence has made a substantial difference. Through the collaboration between INET-Wild and the Agouti project, which was originally developed at Wageningen University, and that is now part of the INET-Wild consortium, we are now able to use an IE-powered platform that is open and openly accessible to everyone with a simple log registration format. The platforms performs much of the images processing workflow using several different models. I report here two of the models that are currently available. The Europe V5 is the latest, most advanced model that is able to recognize over 100 European species, and a model that was originally developed by French institutes that is specifically targeted for 28 of the most common wildlife species present in France. Both models, of course, can be used interchangeably and have been developed keeping in mind the European wildlife focus. What does Agouti do? Agouti does a lot of things automatically that is mainly used for creating and constructing images sequencing. From these constructed sequences, Agouti is able to classify the species, each species that appears inside the image. Agouti can classify it. It can remove blank images, vehicles, any pictures that includes humans. This is particularly important for privacy-related issues, as you know. If any of you has ever worked with camera traps, knows that even filtering the blanks images can consume an enormous amount of time. So automatizing this step drastically increased efficiency and scalability for integrated wildlife health surveillance. However, as we say, is not all gold what glitters, because Agouti cannot do everything automatically. In the RAM workflow, estimating travel distance requires currently that an operator manually identifies the 4 foot of each animal in each frame. As I said, this step is currently performed manually by trained operators. Colleagues from Wageningen are now working to automate this final component by developing an IE model capable of detecting and tracking the front foot across different images. We are not there yet, but this is the next step toward a fully automated density estimation. So while IE has significantly streamlined the process, still expert supervision and manual validations remain essential. To go to the applicable part of what I'm telling you today, how can camera traps data be used to improve surveillance? Let's say the most immediate and intuitive application is for wildlife population monitoring. For example, in the slide here, you can see data camera traps data deriving from an African swine fever outbreak that we had a few years ago in northern Italy, where we implemented an integrated monitoring approach. Using camera traps data, we monitor the changes in population density over time in response to the outbreak, not only for the wild boar, which is of course the target species, but for the entire wildlife community that uses the same area that was affected with this disease. This information is, of course, valuable for wildlife management, as density trend can help evaluate the impact of disease and, most importantly, the effectiveness of control measures. From a management point of view, camera traps can also be used to reconstruct, for example, the daily activity pattern. If we want to know when one species is most active, camera traps can give us this information, and this is particularly valuable, for example, if I have to say to hunters that are in charge of controlling a population, when is the time of the day where they have the highest possibility of actually seeing the animals active and thus to have a fruitful hunting session. Camera traps also allow monitoring of population structure, such as, for example, the proportion of females, of juveniles, of reproductive groups. These are all very valuable information for a management perspective. And of course, they can be used very effectively for detecting the expansion of invasive alien species. From an epidemiological point of view, camera traps give us a lot of invaluable information. Here I present a very broad overview of some projects that we are currently developing within the European Observatory of Wildlife, which is a network of, as we speak, 105 study areas across Europe, where standardized protocols for density estimation are implemented in an harmonized way and coupled with harmonized protocols for wildlife surveillance. A first example on how this integrated approach can be applied comes from, these data are from a study area in Italy and have as target the interaction between density, presence of wildlife species, and abundance of ticks and, of course, of tick-borne pathogens. This study was carried out along an entire year, where we coupled information on temporal occupancy, so the time spent spent by animals of a single species in front of a camera trap with abundance data of ticks, in particular of Ixodes ricinus. Tick abundance was measured through standardized dragging in the immediate surrounding of the camera traps, and of course, temporal occupancy was measured with camera traps. What did we observe? The results clearly showed species-specific effects. For ruminant ungulates, such as the red deer and fallow deer, specifically for this study area, we observed an increased temporal occupancy. We observed how higher temporal occupancy was positively associated with higher abundance of Ixodes ricinus. In other words, the more time these species spent in front of the camera, the more ticks we collected in that area. In contrast, the relationship was reversed for wild boar. Other examples come from, examples of how this harmonized integrated system can be applied come from pilot studies that are currently being developed that have as objects toxoplasma, fasciola, or fascioloides magna. In the next session, my colleague Amir will provide a detailed overview of how, for example, environmental DNA can be used in these integrated pilot studies. However, integrated monitoring can also rely on more traditional protocols that include active and passive approaches, for example, sampling animals harvested during routine cullings or hunting activities. One example comes from this study where toxoplasma seroprevalence in wild boar was connected to wild boar density itself, to density, presence and density of the definitive host, so of the cats, and of course to other known factors such as a anthropized environment that are known to influence toxoplasma transmission routes. To conclude, what are the next steps? As we saw, we clearly recognize the invaluable contribution that IE has made in streamlining the processing and enabling large-scale standardized wildlife monitoring. However, I would like to leave you with a question that is also one of the key directions in our future research. So what if IE could reliably recognize individual animals? Up to now, we use models that are able to recognize individuals at species level, but being able to individually recognize individual animals. This is, for example, something that we are working on with chamois or with ibexes, that with the horn are particularly prone for individual recognition. So what if, this is a big what if, but what if individual recognition would allow would become a reality? Well, this would allow the application of statistical approaches that are based on contact networks, on movement trajectories, and repeated interaction between known individuals. This, of course, will significantly strengthen our ability to reconstruct transmission pathways and quantify contact structures and model disease spread within wildlife populations. I thank you for your attention, and I would like also to thank all the persons that are reported here in this picture that are the true authors of the presentations of today.
Wow, great, Stefania. You really impressed us and showed how parasitology can be very much integrated into ecology. So thanks a lot.
Thank you.
And now we open the discussion for the three speakers. So please go ahead. Is there any question? I can start with the Noemi. It's just a curiosity. Is it relevant the way you place the mosquito into the different slot, or can be anyway?
No, you can be anyway, because the training is made to identify a real situation. So the mosquito is put on ventral position, dorsal position, and obviously the profile is all position.
So this is very convenient for field the situation, I mean. Yeah.
Sì, exactly.
Okay, thank you.
Thank you so much.
Any others?
Yes, I would like to.
Lavinia,
Yes.
go ahead.
Yeah, sorry. I will just open my camera. So I have some curiosity for Cecile's presentation. Yeah. So how much of the quality and also of the size of the reference database could let's say, influence the accuracy of the mosquito identification? And then if this technology is enough to be implemented at the scale large, let's say, entomological programs like surveillance.
Thank you. Yes, it's a good question. We need databases with a huge diversity if we want them to be accurate enough. And also the preservation mode is important, especially for constructing reference databases. So we both need huge diversity and references of good quality, and with an identification with a reference, a gold standard, for example, molecular identification, and combined with morphology. So it's very important to build accurate databases. And for now, we need collaboration to really expand the use of the tool, because we have several databases of mosquitoes from Africa and from Europe, but we are starting and we need an important effort.
Yes, thank you. And how about the simultaneously detection of the mosquito species, but also the pathogen within the same sample? Do you think that, I think there are still things to update, starting with the reference database that you said, but there are current limitations about that?
For now, for the infection, we have only a proof of concept with the laboratory colonies. So we do not know, there are many things that we don't know yet. For example, the specificity of the detection. Are we detecting a signal of the pathogen or a immune response of the vector to the presence of the pathogen? And it is more likely that we detect a immune response, so that this detection is not specific to a pathogen. But we need further experiment to confirm this hypothesis. So for now, we are at the very beginning of the detection of the infection in the vectors, and we need further studies.
Thank you, Cecile.
Thank you.
Are there any other questions?
Hi.
Hi.
After I have a question for Noemi. Noemi, congratulations for your presentation and for the model that you use. I have a question. How many images are needed to have a basic identification for each mosquito species?
Okay, thank you. The minimum is 1,000. 1,000, but is really is a minimum, okay? Because the minimum and the maximums depend on your biological question. In our case, the minimum is more high because we have 7 species and more of them are similar, no? Aedes aegypti and Aedes arbovitus are more similar. But the important question is to out the type of the image, not the quantity, because it's case sensitive, the deep learning model. So if you have high quality, you can also use less image. If you have bad quality, is the number of image increased. Obviously, this okay, but the deep learning model is more different respect the machine learning because the deep learning model increase like an exponential way. So is able to apprehend to the knowledge about the mosquito in this case is exponential because you don't have a plate, okay, the respect to the machine learning. So this prototype can grow in an exponential way.
Okay, thank you.
You're welcome.
Thank you. Other questions?
Maybe I can ask something to Stefania. Stefania, so this Agouti is free of charge? And then at the beginning of your presentation, you mentioned something about the sites of the mammals. So yeah, can you
Yes,
say
absolutely,
more?
absolutely. So
Yeah.
yes, Agouti is a platform that is currently free for anybody who wants to use it. The only constraint is the adherence to the protocol. I mean, of course, you can use the tool, but in order to have access to the full version of it, you have to comply with what is the standard protocol for the EOW. So this is one limitation. And instead, regarding the size of the mammals, yes, this is a constraint related to the hardware part of the camera traps. Of course, camera traps work in detecting difference in temperature of body size. So a very small animal cannot be reliably detected. So let's say the model that we developed is feasible from the size of a lagomorph up. Rodents cannot be assessed with
So
reliability.
maybe you know that we are working here also with Gerard on Echinococcus granulosus, and so stray dogs are
Absolutely. So
yeah,
dogs is
okay.
perfect. Dogs
So
is
I will
perfect.
reach
Yeah.
you very soon. Okay, thank you.
Stefania, I also have a curiosity. Maybe I didn't get everything you you explained, but you spoke about tick density detection, I think. Can you identify the tick species with your model?
No, unfortunately, the model only is aimed for mammals. So the ticks was identified with the stereo microscope. No, absolutely not. No.
So you just can say, okay, there are ticks on the map.
The automatization part of the study that I showed relies only into what concern the host species, not the vector.
Okay, thank you.
Great. Bravi, bravi. So Greg, I think we can move on the next session on eDNA. And it's my pleasure to introduce Olivier Rey from the University of Perpignan and he will talk about environmental eDNA and eRNA for parasite monitoring insights from trematodes.
All right, thanks very much.
welcome.
Good afternoon, everyone. So let me just share first. I'm not sure about which will be the best. No, probably not.
Yeah, we can see.
But you have
we
the
see
wrong, right?
yeah.
So let's sorry. Is it better? No, not still. Okay. What are you seeing right now?
No, we see also the second one, but
on.
maybe you can put on you can click on the top left if you put
left.
maybe the second,
Here.
You put presentation and then on the top left.
It should be fine.
Okay, perfect.
Okay, sorry about that. Sorry about that. These technical problems.
No problem.
So thanks very much for giving me the opportunity to present some studies that we are currently doing at the IHPE. So my name is Olivier Ray. I'm an assistant professor. I'm a molecular ecologist, and I will show you some studies that have been mainly done by all these students that you can see here: Stéphane, Philippe, Mathilde, and Chiara. But maybe just before starting, I just want to introduce a little bit the lab. So we are the Interactions Host Pathogen Environment Laboratory, which is an integrative lab, which is led by Christophe Bruno. And we work on interactions, of course, between pathogen host and environments, from molecules to the ecosystems. And I'm mainly involved in what happens at the ecosystem level. And we share some different models, common models, biological models, including trematodes and their hosts, but also microorganisms with bivalves and so on. Also working on corals. I will just speak about trematodes today. And one of the questions that I'm trying to answer is how global change influenced the transmission of trematodes. Why this question? Well, you probably know that trematodes are parasites that display complex life cycles. This is just illustrated here with a life cycle of schistosoma species, where organisms shift from definitive host to intermediate host, where sexual reproduction occur in definitive host, asexual multiplication occur in the first intermediate host. Which is important is also that all first intermediate host, whatever the trematode species, is always mollusk. So it's important to target these organisms when we are interested in the transmission ecology of these organisms. And just very quickly, also briefly to end the presentation of these organisms, you might know for at least these species that parasite goes from definitive host to first intermediate host with free living stages, what we call it Miracidia, and from the first intermediate host to the definitive host with Cercaria. This will have some importance later on. And this is why we might expect that the transmission of these organisms is very influenced by the environment, by biotic and abiotic factors. And this is where I'm interested in how the changes that occurs at the biotic factors, or even on the biodiversity overall, the human impact the transmission of these species. So I use concepts, laboratory experiments, field studies, but all along my studies, we definitely need to develop some molecular tools. And the one that are of interest today are the one that they stick on environmental DNA, which is here a definition by Pierre Taberlet in 2020, which is a complex mixture of genomic DNA from many different organisms found in a given sample, environmental sample. And here we define environmental sample as different matrices. It can be water, it can be sediments, it can be feces, it can be also snails when we look at trematodes inside the snails, or microorganisms such as bacteria and so on. So maybe one first interesting thing in environmental DNA is to target some specific intermediate host species to identify potential sites of transmission, given that this is like the door for trematodes to enter into a system. So if you if you can detect or identify the presence of a given host, you might expect that transmission can occur for a given parasite here. So this is what we've done with Stéphane Mulero using digital PCR technology to target Bulinus truncatus, which is the host for Schistosoma hematodium and Schistosoma bovis. As you can see here, it is also interesting because we can quantify the host abundance. So here is semi-quantitatively, but we know now that it works very well to have an idea of the abundance of the hosts in a given environment. Instead of looking at just one snail host, you can look at the community of the snails. So it can give you the potentiality for other trematodes to enter to the system. And it also allows to study different ecological processes through which the transmission of some trematodes can be impacted, such as snails competition or dilution effect and so on. So here is the results that Stéphane had during his PhD, so it works quite well. You can see here, so this was a malacological survey that had been done in Corsica at 23 sites. And at the same 23 sites, we looked at the community of snails, using environmental DNA monitoring. And as you can see, the results are very, very similar, which means that it worked quite well, and we can have an idea of mapping of each snail species at different sites. Instead of looking at the snails, you can also look at the parasite communities, here trematode communities. So this is something that Philippe developed, a tool to be able to characterize all trematode communities from the environment, from environmental samples. So this is a first study that we've done a few years ago, where we compared the communities of trematodes characterized through environmental DNA compared to classical sampling, where we collect snails, we let them emitting some Cercaria, and we can then look at which Cercaria, which species is. And as you can see here, we identified 8 species only with environmental DNA, 3 that were common with the different techniques, and 2 only with classical sampling. So it works quite well. It gives a better idea of which trematodes are circulating in a given environment. More recently, this tool also allowed us to monitor the dynamics of trematode communities in natural reserve, the Bagnas Nature Reserve, which is in southern France. And as you can see here, which is on the way of a lot of different migratory birds. It's a very known stopover for birds. And as you can see here, the community of trematodes, or the complexity of the community of trematodes, follows the specific richness of birds, of migratory birds during time. It works also not only for the environment, as I said before, we can also use these kind of tools to see what happens in snail hosts. So this is a study that have been carried out in Senegal, where we found 20 species of trematodes living in these different areas from environmental DNA. 9 of which were found with Bulinus truncatus. And when we make the metabarcoding on snails, we found that some of these snails could be sometimes co-infected by several species of trematodes, up to 4 species within a given snail. So these this result gave us the opportunity to study a little bit more what happens in terms of interactions between trematodes within snails and how this can affect the transmission of, in this case, specific case here, schistosoma hematoma. Instead of looking at all the trematode communities, of course, environmental DNA can also be important to target some specific trematode species to identify active sites of transmission and quantify the parasite abundance in the environment. So as you can see here, there are different examples. We are not, of course, alone in working in this kind of approaches to monitor parasites. But here, one limitation I would say is that we don't really know what we are currently quantifying when we use environmental DNA, because in the case, for example, of Schistosoma mansoni, Cercaria, Miracidia, eggs, and so on will have exactly the same DNA. So we can't know, well, we don't know what are we really quantifying when we identify the presence of a given parasite. It can be also, as I said, DNA from eggs or even from dead organisms that remains in the environment. So one approach is to shift from environmental DNA to environmental RNA, and to use the fact that each of these life stages provide some specific transcripts that can be used to be targeted and to better quantify what happens in the water. So this is something that we started to work with Philip, and now is part of Chiara's thesis, sorry. So these are the results. So first, the main idea was to find these kind of transcripts that are specifically expressed by Miracidia in the case of Schistosoma mansoni again, and those that are specifically expressed by Cercaria. So this was done using the Schisto XYZ atlases database, which is online. And from this, we developed some primers and some probes to use them with digital droplet PCR. So here are some results that we obtained, so that Chiara obtained, to be more precise. So what we can see here is that the probes, sorry, are very specific to Schistosoma mansoni, because when we put some mock communities with Schistosoma mansoni, it works. This is a positive here, sorry, a positive sample. And when the same mock community is used without Schistosoma mansoni, there is nothing going, well, nothing is positive, the negative control is negative. So we definitely have something which is specific to Schistosoma mansoni. We also have some here, so two different probes, one which is blue, the other green. The blue one, which is specific for Miracidia, the green one, which is specific for Cercaria. And we see that there is very, very little cross-amplification from one targeted RNA to the other. So something which is also very specific to each life stages of Schistosoma mansoni. And as you can see here, but we will see it later on, it also provides, well, we should expect that it is important to quantify absolutely the quantity of Miracidia and Cercaria from environmental samples. So the next step was to try with environmental RNA, sorry, but in controlled laboratory experiments. So what we've done is to put some Miracidia and Cercaria either at the same ratio or with unbalanced ratios. And we filtrated this extracted RNA and DNA and looking at what happens. So this is the first result, which is important for the same ratio of Miracidia and Cercaria. You can see that we can definitely well detect the presence of Cercaria or Miracidia in the water. We better detect Cercaria than Miracidia, and we better detect RNA from Schistosoma mansoni than DNA. And then when we look at what happens in the unbalanced treatment, well, what we can see here is that the observed values of the quantity of Miracidia or Cercaria RNA stick very well with what is expected under what have been computed at the 25 ratio of Miracidia Cercaria. So we here again have a proof that it works quite well to accurately quantify the number of Miracidia or Cercaria that are in the water. The next step that we wanted to check was to check the longevity of environmental DNA and environmental RNA. So this we've done that with following what was happening in the water at day 0, 1, 2, 7, and 12. And we looked at what happened for RNA and for DNA. And as you can see here, even if everything decreases at day 2, you can still find some RNA and some DNA. At day 7, almost everything has disappeared, even if we can still detect RNA. So this means that we are not able to differentiate between living and dead organisms, because 2 days after, it's much after the lifespan of Miracidia and Cercaria. But the good news is that the efficiency of RNA still works a few days after the organisms are dead. And finally, what we wanted to see is if we were able to detect free environmental RNA, which means when we remove the organisms first before filtering water with our sample. And these are the results. So as you can see here, when there are some organisms, the quantity of Cercaria RNA, Miracidia RNA, and DNA is very high, but we still are able to detect something even when we remove the organisms just before looking at what happens in terms of environmental and RNA and DNA. Sorry. And at the end, I'm going very quickly. I'm almost out of time. We've done a field survey in Brazil, where we knew that at some sites, Schistosoma mansoni were circulating. We've been at 6 different sites. We collected some snails, let them emitting, and made the eDNA environmental RNA diagnostic. What we found is that at 2 different sites, we found some snails shedding some Cercaria. And at these exactly same sites, we found some environmental DNA. We found environmental RNA only for Cercaria. We did not found any environmental RNA from Miracidia, which is quite expected, I would say. So just to end up here the presentation, the usefulness of environmental nucleic acids, either environmental DNA or environmental RNA, is important to detect hosts, either only one species or multiple species. When we are looking just at one species, we can quantify also the abundance of the species. Same for tomatoes. We can have an idea of what happens in terms of communities in the environment or in the hosts, and all this gives some information about the potential for the active site of transmission. And then environmental RNA is a very interesting tool to tease apart what are we looking at, either Miracidia or Cercaria in the case of Schistosoma mansoni. So it gives some information about the presence of contaminated host communities, either humans or animal reservoirs, and also give a better assessment of risk of transmission of these parasites to human patients. And everything also gives a better idea of how transmission occurs in the field. So thanks very much. Sorry for being a little bit late. And I'd be happy to hear about your questions if you have.
Thank you, Olivier. Thank you. And we will have our discussion at the end of the session. So, Greg, you want to introduce the next speaker?
Yes.
No.
And I want to thank you, Olivier, for agreeing to present this very interesting presentation. And now I will ask Amir Reza Balzandi from the Department of Veterinary Science at the University of Turin. He will present the monitoring environmentally mediated parasitic diseases using environmental nucleic acids. Amir?
Hello. I think you hear me? Yes.
Yes.
I hope. Very good. I'm going to share the presentation.
Okay, thank you.
Okay, I hope you see the presentation.
Yes.
So I'm going to talk about, thank you for the opportunity actually, and I'm going to talk about the monitoring environmentally mediated parasitic diseases using environmental nucleic acids, particularly environmental DNA. A brief background about myself. I'm Iranian. I have studied animal sciences in Iran. And then I studied animal biotechnology master's course in the University of Bologna. I worked many years in pharmaceutical companies, but I finally returned back to Italy and pursued my PhD in veterinary science under the supervision of Prof. Ferrolio. And nowadays I'm collaborating with Prof. Ferrolio and Professoressa Zanet in the Department of Veterinary Sciences Parasitology section. Today I'm going to talk about environmentally mediated diseases, or diseases that can persist in the environment, remain infectious for more than one day. They are very important, particularly for the wildlife, as they are reservoirs for many of these diseases. And they are in contact with the most persistent diseases. Can be bacterial, can be parasitic diseases. They can affect animal health, human health, or wildlife conservation. So they are very important to be surveyed, to be monitored, especially to be detected as early as possible to avoid any emerging epidemiological situation. They are, as maybe you remember, Stefania also mentioned, there are many methods to monitor wildlife or the pathogens like capture, mark, recapture, camera traps, and many other, but they are very cumbersome and labor intensive. Nowadays, environmental DNA promises a very easier way to monitor wildlife communities, animals, and in total environment. If we consider environment as a container, using environmental DNA, which can we can assume that it represents what organisms is in that environment, can potentially find different hosts, pathogens, or vectors. Our container in this study is our study area, La Mandria Regional Park, which is close to Turin. It is interesting study area, which is divided in two areas with a fence, and inside it has separated with a fence, which is made of a wall, actually. It includes tributary rivers to the Po riverine system and hosts 4 species of wild ungulates inside the fenced area. And it is now to be the first European focus of fasciolus mania, a term that our colleague before me also explained well about the complex life cycle that they have. Fasciolus mania actually has the white cervix as its final hosts. It needs water for its eggs to be hatched to the Miracidia, and Miracidia needs snails, limnated snails, actually dextrolimnated snails, to complete its life cycle to become a metacercaria, and metacercaria is placed by the snails on the top of the grass, which is finally ingested by the final host. What we did at first in this container, in this study area, was to develop, design a qPCR assay detecting both the pathogen and the parasite, Fasciolus mania, and the Galbatroncutula, validated in the laboratory using the water tanks, mesocosm studies, and then collect samples from water, whether inside or outside the fenced area, using an ad hoc sampling strategy. First, we demonstrated the specificity of our assay, established the limit of detection, and we also control with the water tank studies how is it feasible to, is it working, and validate our test by extracting DNA from these water tanks in 3 different time points up to 2 weeks. Then we went in natura, in the park, in our container. We found places that eliminated snails, the externally eliminated snails were present or not, and collected water, filtered water with a capsule filters, and then tested the extracted DNA out of these filters with qPCR developed by ourselves. And what was interesting that we found 2 points immediately upstream and downstream of the fenced-off area to be positive for the fasciolus mania, which are actually outside of the reach of the wide ungulates or final hosts. And the one that is in the upstream can be assumed to be representative of DNA that is coming from the much more farther away. But it is not enough. We wanted to go for the early detection, to go more and search for what is not expected with the environmental DNA. Using the nanopore sequencing, which is a portable device, it enables us with a PCR-free and direct sequencing. It sequences long reads. It also enables us to do adaptive sampling, which is a kind of a priori in silico enrichment or depletion. And it also at the same time captures the genetic data of the sequenced fragment of the DNA. We analyzed the same samples we collected from the last study, previous study. We pulled those replicates together and used those pools to sequence DNA with the nanopore native sequencing kit, which avoids fragmentation of DNA, and directly sequence the DNA as it is in the sample to find what we can find actually. What is important with the native sequencing is that we had a lot of reads, but only a minority of the reads were classified. We used 2 classifiers, Kraken2 and Megablast. And inside the classified reads, the majority belong to the bacteria. However, in the least abundant reads that we found, we were able to find again fasciolus mania, Cervus elaphus, and unexpectedly, we also found Batrachochytridium dendrobatidis. These samples come from the September 2022, but in the same park, in the same container, 6 months later, we had an outbreak of Batrachochytridium dendrobatidis, which caused a high mortality in the amphibians. It was very interesting to find it even as early as 6 months before the cases being reported. But we were not able to find the Galba truncatula, the host of the intermediate host of the fasciolus mania, which we previously found in our last study. We checked our samples with ddPCR for the Batrachochytridium dendrobatidis, and we found some positive samples, positive droplets in some of our samples too, as you can see. Next step for us studying our study area using the environmental DNA was to consider that if we consider our study area as a container, how can we find places to go for the sample collection, which represents the major biodiversity in that container? So, assuming this hypothesis, we found or identified different water basins and flow segments, and defined beginning or the end or joints of these water segments, like point 1, which is the beginning of a water segment, and point 5, which is a joint place as 2 flow segments from point 3 and 4 arrive, and selected these points as a kind of as an effort to go for standardizing the places we go for the sample collection, which are represent much of the biodiversity in our container. So finding those places, we combined the sample collection from water and soil, tracing the runoff we collected soil. At the end of the runoff, if the water, stagnant water was present, we also collected the water. Then we extracted water and soil from our samples. We first did qPCR using our duplex assay, but we didn't find anything. It was all of them almost were negative. So we moved forward using the ddPCR, and we found positive samples using the same samples after testing with ddPCR. Actually, our sample collection was for 3 months. Every month, same places, water and soil were collected, extracted, and as you can see, that the results from the first month, that was August, were showed to be more positive results, to have more positive results. And by the time in September and October, the number of the positive results for both galba truncatula and fascia mania decreases. This decrease is in line with the overall trend observed for the galba truncatula, because the majority of the highest population peak for this limnated snail is in the summer, here in August, and it decreases by the time as we go and get closer to the winter. What we can see is actually that we were able to demonstrate that the qPCR is working. Our qPCR is working on the environmental samples collected from Lamandria Park for the detection of galba truncatula and fascia mania. We also, on the same samples, demonstrated that nanopore sequencing, native sequencing, also has, although has many challenges still to be overcome, can find the unexpected and also find the things that the species that we found with the qPCR, and that the combination of sampling strategy, a much more systematic sampling strategy with PCR-free sequencing can be very promising for eDNA-based pathogen monitoring. We also detected many challenges, as also was mentioned with our with previous colleague, that we cannot find with the eDNA directly the state or the stage of the DNA that we find. Actually, it is very important in the DNA in pathogen detection for the parasites. It is important the DNA that is released and is found in which belongs to the which stage of the life cycle of the pathogen. With nanopore, we are still able to go and search for this with the methylation calling. And nowadays we are trying to, with sequencing the different stages of the parasite, we are trying to find the differences in the methylation patterns of the stages of the life cycle of the parasite, so that the same DNA that is sequenced can be also used for the epigenetic data to see in which life stage belongs to. And also we are trying to standardize more our sampling strategy and in order to place and position eDNA in more integrated wildlife monitoring systems. Thank you for your attention.
Thank you very much.
Thank you. So, Greg, you can introduce the last speaker.
Yeah, sure. So
Good evening.
Yeah, Maria Letizia Palomba from the Department of Ecological and Biological Science of Tuskegee University. And you will present the eDNA-based approaches for monitoring aquatic metazoan parasites. So,
Yeah, Emma, Letizia is also the youngest, I guess. So good luck, Letizia. We are with you.
Thank you. Good evening to all.
Hello, I'm Maria Letizia Palomba from University of Tuscia. I'm tenure track researcher, and it's a pleasure to be here and present our preliminary research on environmental DNA-based approach for monitoring aquatic metazoan parasite. But why? Sorry, but I have a problem. Okay, but. Sorry, I'm not able to see the presentation. Okay, but just a short introduction about why eDNA for aquatic metazoan parasite. Like we know, parasites are key component of biodiversity and ecosystem functioning, but monitoring aquatic metazoan parasite, in particular marine parasite, is often challenging because detection typically requires host sampling and free living stages are difficult to observe. So environmental DNA can provide a non-invasive detection tool that offers the possibility to detect parasite presence directly from water, including life stages. So we have two main eDNA detection strategies. The first one with real-time PCR that permit to detect specific parasite, one or more, with high sensitivity, and next generation sequencing that is also called metabarcoding, that provide a broad overview of biodiversity, including parasite and host. So the aim of this presentation is to present two eDNA-based approach for monitoring aquatic and in particular marine parasite system. So from a side we have the real-time PCR that permit a species-specific detection approach targeting in particular two sibling species of Contrachecum rudolfi complex using a diagnostic marker, and from another side we have a community-level approach based on koi metabarcoding in order to explore host-parasite interaction between Polydora species and oyster. eDNA can be applied at different levels from single species detection to community-wide analysis, but the whole depends on our question. So the first case study is about Contrachecum rudolfi species complex. We focus on Contrachecum rudolfi and in particular the sibling species Contrachecum rudolfi A and Contrachecum rudolfi B from European water that are anisakid nematodes with complex life cycle involving fish eating bird as cormorant as definitive host and first intermediate host as a small crustacean and teleost fish as intermediate host. The objective of this study is to assess the environmental presence of the two sibling species in order to explore the potential ecological preferences. On a previous experimental studies on egg hatching, we suggest that Contrachecum rudolfi species A, as we can see in this graph, tolerate higher salinity, while Contrachecum rudolfi species B is mainly associated with lower salinity condition. So the first step was develop a species-specific assay based on real-time PCR. So we select a diagnostic position in ITS gene and we design a primer and probe specific for Contrachecum rudolfi species A and B. After then, we validate the primers and probe in laboratory on adult parasites that were before identified with a Sanger methods, and after then we design a standard curve that permit to observe a high specificity, sensitivity, and amplification efficiency across several dilutions. After then, we evaluate assay performance under controlled aquarium condition containing, as we can see here, increasing larval number from 2 to 50 L2 in each aquarium and a separate aquarium with eggs. Each aquarium from each parasite. So we have Contrachecum rudolfi species A, B, and the combination of two. Also, negative control were included in the experiment. The results show high sensitivity across larval abundance, like we can see here, and we can see that eggs are able to release detectable DNA. After that, we sample water from different coastal and freshwater systems, in particular Orbetello Lagoon, Fiora and Marta River, Tarquinia Salten, and Paula Lake. After then, the water was filtered and DNA was extracted and tested in real-time PCR for both species, Contrachecum rudolphus species A and B. The results show the presence in all sites of Contrachecum rudolfi with a differential distribution across brackish and freshwater systems. In particular, Contrachecum rudolfi species A was detected predominantly in brackish coastal lagoon, while species B was detected mainly in freshwater system, with a clear habitat-associated spatial differentiation. The second aim was to explore host-parasite interaction between Polydora species and oyster with a community-level approach based on koi gene. Just a short introduction about parasite. Polydora species are shell-boring polychaete worms living within bivalve shells. Adult inhabit galleries inside the shell where they feed and reproduce. They provoke a bad-smelling mud blister that reduce hoster marker value. A planktonic stage is also present that facilitates dispersal and potential spread. So the objective of this study is to assess the possible presence of Polydora species potentially introduced through the no introduction of Pacific oyster Magellana gigas in the study area. But in general, what happens to parasites during a species introduction? We can see three different scenarios. An animal release where invasive species may leave their parasite behind when entering in a new environment. A parasite spillover where parasite introduced with invasive host can infect native species, and a parasite spillback where invasive host can amplify native parasite and increase transmission. So within the framework of a national project, a PNRR project, several coastal lakes along the Lachin coast were sampled, in particular Fogliano Lake, Monaci Lake, Caprolace Lake, and Sabaudia Lagoon. Water samples were filtered and DNA was extracted, and amplification and sequencing of koi gene was performed. Sequence data were then processed with a specific bioinformatic pipeline. The result show, as we can see here, both the introduced Pacific oyster Magellana gigas and the native oyster stentina were detected across most sampling sites. In addition, both the native, that is Polydora oplura, and the introduced species Polydora neocerca and Polydora websteri were detected, and only in sites where both oyster species occurred. So, two possible scenarios are here. The detection of potential introduced Polydora species is consistent with a spillover scenario where parasites are transported together with invasive hosts, and the presence of native Polydora species that may reflect spillback dynamic where invasive hosts can contribute to parasite amplification. So, in conclusion, environmental DNA provide powerful tool for monitoring aquatic metazoan parasite across different ecological scales. From a side, we have the real-time PCR that allow precise detection of cryptic species and reveal fish-scale ecological differentiation. From another side, we have the meta barcoding that permit to expand our perspective to community-level dynamic, a potential invasion-mediated process. Together, this approach demonstrates that parasites can be monitored not only through their host, but directly at the ecosystem level. So thanks for attention.
Thank you very much.
Thank you. Thank you,
Thank you to
Letizia.
you.
And thank you all the three speakers for this very promising tool you developed all in your labs. And now we have time for questions, discussion.
I
Yes.
have a question.
Yes, Ines.
Okay. First, I would like to thank all the presenters for their interesting and valuable contributions. Then my question is as follows. It's about details for the methodology that I didn't fully understand. During the water sampling, how much the quantity of water my colleagues collected, and they said that the water was filtered. Did they proceed directly to the DNA extraction, or they keep the filter in some solution, then they extract the DNA? And also, if they have a big site, like, I don't know, lake or a river, did they make sampling from different point in these sites, or just from one point it's enough to detect the infections? And thank you so much for my curiosity.
Crucial question. Who want to answer?
Can I start just because we had different types of sampling from water. In our first study, we used the capsule filters, and we didn't have any limitation for the filtering, because we filtered water up to the clogging of the filter. And the minimum amount of the water that we filtered was 3 liters. And we also, in some places, filtered up to 20 liters. But in the second study, because this is it is right that it's a non-invasive way of sampling, but still it requires a lot of effort. So in the second sampling effort, we only collected 200 milliliter of water for falcons of 50 milliliter, but our molecular method was highly sensitive, because we used ddPCR. So we assume that if we reduce the sampling amount of the sample of water we collect, we can compensate it with higher sensitivity molecular methods. The water that we collected in the falcons, then in the laboratory, we filtered again, and we extracted DNA from the filters.
Okay. Okay, thank you. Okay.
Yeah, just maybe one adding about what he said. So we tested also some different ways to collect environmental acid, nucleic acid, from the environment. We started with flat membranes, as some colleagues have done also, where we usually filtered as up to 3 liters. Then we shift to the capsules, where we can filter up to 40 liters. And because what we're looking is very rare, we were thinking about making like a first filtration with using plankton nets, and to optimize the amount of DNA from these parasites. And we've done a full experiment on using plankton net or capsule. So plankton net, we estimate that we collect up to 500 liters of water, but it did not improve at all the results based on metabarcoding. So it seems that there is no need to try to collect more and more water, but maybe, as you said, to collect at different points or even at different time scales, because parasites are not released at the same time a day. So I would say that the best solution would be maybe to try during time.
Okay, thank you. Thank you.
I just saw that there is a question in the chat from Barbara Heaney. So Barbara, I'm happy you could join us today. And so she's asking, because they sampled eDNA in water for testing for fasciola, and the qPCR was negative. And so she's asking, could it be that it would be positive in ddPCR? Can someone answer, or maybe we cannot answer, I don't know.
Can I think yes, if we assume that ddPCR is, we had this experience actually. With qPCR, we didn't find a lot of positive samples, so we moved on using ddPCR, and we found low quantity positive samples, but they were positive.
Thank you very much, because we did monthly samplings over a whole year, and we know that there was fasciola, and we even had snails, but all samples were negative. So perhaps that would be a solution to test the samples again with digital PCR. Thank you very much. Yeah.
Fasciola are not very friendly because they metasaccharize. You might assume that they not release that much of DNA. So once they are metasaccharized, it's much difficult, I would say, to connect. But it has never been tested empirically. So this needs to be tested, definitely.
Okay, thanks. You are cool that I could join here, because you're really experts in this field. Yeah.
Olivier and maybe Amir and Letizia, do you think for fasciola specifically we could also maybe try on vegetables, because the cyst is forming in the sheet of the vegetables? So
Yes, I think that is very similar procedure. So yes, we have to try.
okay.
I have just a curiosity. So why, according to you, most of the studies have been done on trematodes? Because of the life cycle, the different, and what about other parasites?
So, no, I have plenty of examples of different parasites which are not trematodes also. I think some of them are not really well studied right now, but it's more a problem of methodology. For example, for nematodes, I know that it's very difficult to get only one metabarcoding because of the genetic resolution. So it makes it a bit more difficult. But then I have some other example of monogenes, for example, or I can't remember this one, the proliferative kidney disease of salmonids.
Okay, so there are a few other examples.
There are a few that works, but yeah.
But maybe the methodology has been well established for better established for trematodes.
For example, in our case, we have nematodes, so we have not problem about this, but we used mainly the real-time PCR and not metabarcoding technique. So it's a problem for the primers, the universal primers. So in real time, it's good for the moment.
And
we also, excuse me, we also use, if it is targeted, different parasites, we are able to find from Toxocara as a nematode or apicomplexans, Toxoplasma. But when it comes to the more universal approach and doing metabarcoding, it becomes a little bit difficult, as it was mentioned.
And my question, Amir, would be, would you tell that nanopore approach, I mean, not targeted approach, would be better to be sure that we find all the parasites which should have been present in the same host, or do you think we should maybe design kind of microfluidic approach to target many parasites? So targeted versus not targeted approach.
Honestly, we are trying to go toward a more holistic approach. I know that targeted approaches like qPCR targeting 1, 2, 3 is very feasible. It is more reliable. So I think what can help us much more in the field of early detection is to search for what is not expected, and not in a passive manner to search for what we suggest to be present. Nanopore can help us having such an approach with native sequencing in a way that we can sequence whatever inside the sample is present. But the problem with nanopore is error rate, it's the depth of sequencing, and it is very, in eDNA camp, we have, in the field of eDNA, we have much more fragmented DNA than the genomic DNA. So it faces a lot of challenges, but it is promising. As I had the experience, we found in at least one of samples, the same result that we found with the qPCR for the Fasciolus mania. And one organism, one pathogen that we were not expecting to see, but it was present and it was confirmed with the ddPCR. It should be repeated many times to be statistically significant, just with one study, but it seems to be promising.
So if it's more fragmented fragments, would you expect that Illumina will be more relevant?
Illumina, you can, it's very difficult to have a comparable approach with Illumina. With Illumina, you can do metabarcoding. I know that nowadays they have some sort of semi-native sequencing platforms, but yes, for sure, with highly fragmented, maybe Illumina is working better, but the approach would be metabarcoding. What we did with native sequencing, nanopore sequencing, is PCR-free. So they are completely different in nature.
And Amir, just to go through a bit more deeper of what Gregory was saying, using shotgun analysis with Illumina couldn't help because there is no PCR.
Yes, but
there is no first step PCR.
It can be at that. Another, I think, what justifies using nanopore sequencing is that, as I told you, we have the methylation data too.
Yeah, that's true.
It is something that is it is making the nanopore sequencing much more interesting for the field of eDNA. It is very hard, but I think it could be a very interesting holistic approach to adopt in the near future. Not exactly all of them right now.
Okay, thank you very much. I see another question in the chat. So Isabel Arzul is asking if testing the presence of parasites in the sediment would increase the chance to detect the parasite.
So yeah, maybe I can answer to this question. So what we usually do when we collect DNA from the environment using capsules, not flat filters, but capsules, is that we try to collect what happens in the water, but up to, well, down to the bottom, and also we collect a little bit of sediments because, as you said, I think you're right. You might increase the chance to detect some trematodes because all the dead parasites will be down. But digging for looking at them too far in the sediments, I would say that you might have the risk of having a signal of what happens at a very long time scale. That's what you're looking at the ecological scale. If you want to look at what happens right now, if you want to get a precise signal of what happens at a precise moment, you might prefer probably water or just the very upper level of sediment.
Okay, thank you. Any other question?
No question.
No question.
So maybe, Greg, I would like to, oh, it's my telephone, sorry, I have some problem at home, but it's okay. Yeah, I would like to thank all the speakers and the attendees. And yeah, I think, Greg, we really built up a very good, innovative webinar. And I'm sure we will do the same next year or even before. And I would like to remind all the participants about our conference. Maybe, Greg, you can start because you have first your conference.
Yes, we have our conference very soon because it's at the end of April. So from the 28th to 30, because it's joint meeting with the French Society of Parasitology and also the French Society of Medical Mycology. And this year, it's each 3 years, we also have the Maghrebin Society of Parasitomycology, the 3 Maghrebin ones. So, but still, there will be, no, it's opened. The call for abstract is opened, and it will be closed in the 15th of March. So you still have slots, I think, to present some works. And I would be happy if some of you can come. So yeah, it will be my pleasure.
And we also have our biannual Congress of the Italian Society of Parasitology. It will be in Bologna from the 23 to the 26th of June. Our colleagues in Bologna has just launched the call for abstracts, and it will be closed the end of March. So you are all invited in Bologna as well.
Okay, thank you very much. And for all of you, there will be also the ICOPA meeting this year in Montreal. It's the 3rd week of August. And I think the call for abstract was extended up to today, but there will be probably an extra call. So the late call, they call that. So probably you can continue to prepare some abstract if you consider attending this meeting in Montreal. And I think it will be very successful. It's very good for networking, and they plan to have 1,500 people coming. So it's very good for networking. And I know our Italian colleague Marco Lal is in the board, such I am. And yeah, we are now preparing the different sessions. So
and also Simonetta Mattiucci is in the board. So we have a strong representative.
Yeah, I saw Simonetta is.
Simonetta should be online. Yeah. Okay, so we are perfectly on time as usual. So, Greg, we are a good team.
We're strong.
Yeah, well done. Grazie a tutti e a presto, buona serata. Thank you.
Grazie, arrivederci.
Merci.
Merci.
Merci beaucoup.
Merci beaucoup. Ciao Simonetta. Ciao.